Wow, this is a great room. Thank you all for being here today. I'm going to talk to you about what we focus on in my group primarily, which is about why the brain is so variable in time. You could say moment-to-moment time-based analysis. So here's an example from a review in 2008 that's arguing basically at every single level of the nervous system, no matter how fine it is. Here's an example of someone catching a ball. That from the sensory to the cellular to the electrical channel to the synapse and onto the muscle spindles, there's incredibly high levels of stochasticity, but yet this person will catch that ball from time to time at least. So it's not a question of whether in neuroscience the levels at which we measure have variability in them. It's a different question to ask whether it's good or not. So that's sort of what we're interested in. And for a while it's been argued that there are many benefits of this temporal variability. For example, computational work showing that networks formed in the presence of variability and noise are more robust to disruption, that a system can explore more neural states, perhaps enhancing its learning and environmental adaptation, providing some kind of system flexibility, if you have it. Yeah? So in our primary interest in human aging, this is an easy fit because the sort of classic notion of age-related neural noise has been around since the 1950s. That cognitive deficits are due to some sort of greater noise or instability in the brain, some SNR reduction. But these early notions didn't have brain in them per se. Right? They were all behaviorally bound. And if we think about these ideas we just briefly mentioned about dynamic range or system flexibility in general, that this variability could rather be the opposite of expectations. Right? So as you get older, this system may just shut down these dynamics rather than the other way around. So a quick snapshot of some things that we've done in the past, and then we'll focus on some relatively recent work afterwards. So what we see since about 2010 is that the younger you are, the faster you are, the more stable your reaction times are, number of different tasks within and between person designs, the more variable your brain is. So young high performers have very dynamic neural signals. And as you age, there's a broad-scale reduction of dynamic range with increasing age, accelerated age, and poorer cognitive performance. This is parametrically modifiable as well. We showed in 17 that using some tight sort of vascular controls, this issue has been of clear importance for us at every stage, that the reason that you have age differences in variability is not just because of vascular rigidity. So we had computer-controlled carbogen-based hypercapnia in one study with more to come, that we cannot fully at least eliminate age differences in signal dynamics once controlling for those things. But today what I want to focus on is neuromodulation, in particular the dopamine system, which we've focused on the last 5 years or so. So older adults exhibit, for a long time argued, lower cognitive deficits due to dopamine system degradation. That's been measured in different ways. And in animals, at least at cross-section, there's about a 10% reduction in receptor density per decade between 20 and 70. Of course, we don't have that information for humans, so we rely on that as a model. So as older adults generally exhibit less signal variability as we see it, it's plausible then that dopamine is a part of this story. So this idea is sort of driven in part by some work by Palladini in a very nice mouse model with dopamine knockout types. So in a normal mouse, what you notice is that there's different types of dynamics that are occurring at the top there. So differences in bursting rates, differences in timing between those bursts or single spikes. And if you knock dopamine completely out of that system, these mouse models have zero endogenous dopamine, at least as they can measure it. And you see a complete absence of this dynamics. It becomes quite deterministic, single spike, lack of bursting. If you give L-DOPA then to those mice, it provides the only source of endogenous dopamine, and they start to recover some aspects of those dynamics, right? So we like to think about this simple mouse model as perhaps a toy model for the aging process as we think about it. So if dopamine is really a part of this, we should be able to see that. So in a first study, we agonized that system through amphetamine in a within-person design placebo and amphetamine on an NBAC task. And we measure bold variability in all these things. And what you notice at placebo is a major age effect, which we always see independent of the task type, with a parametric ramp in the young. On amphetamine, at the group level, there's very little shift in the young adults. And it turns out they're sort of riding at the top of this inverted U. If you're interested in that, please ask. But at the group level on amphetamine, there's a massive shift possible in the older adults. And that shift is primarily happening in interesting regions like the left putamen, the dopaminergic system, and the bilateral DLPFC. So these systems are elevating their variability. When they do that, at least in people that are task naive, people that elevate bulb variability more on the drug are also getting faster in the older group and more consistent in reaction times in the older group. So some evidence, at least within person, in a two-way within design, of course, drug, placebo, and load level, that this is possible to recover. But because we're in a network neuroscience symposium today, I should probably move beyond these voxel measures and get at what we're interested in that regard. And last year, we had a paper kind of thinking about how to embed, looking at local dynamics within the realm of functional integration or connectivity or whatever name you prefer. So there's work in animals by Carandini and Harris at UCL where they argue that most of the spiking variance that you can find is rather attributed to synaptic inputs rather than local randomness. And that's important because then that means the fluctuations that you can measure should be a function of things that are shared, right? So similarly, two neurons, here's a cartoon on the right. At zero, these are neurons that have the same kind of tuning to Gabor's. And you notice that in the noise correlation, so not when they're looking at Gabor's, but just in general, they peak when they are more similarly tuned to do things in general. So we can use that as a model for system integration overall. You know, if you need regions at long range, short range, whatever, to talk to one another, that should drive variability up. Yeah, this is the idea. So Pincus, as well in the early 90s, had this idea about biological systems integration, could show the dynamic range of output at any point in a biological system should decline when you fractionate that system from itself. Yeah? So you're disconnecting these pieces. So another way to look at it is like this. You know, if you had, I guess you can't see the transparency, but region A in the bottom middle, 3 examples of outputs from that region when they have multiple inputs, 1, 2, or 3. And the idea is that if you have more inputs feeding into A, the probability of dynamics in A increases because each of those regions is not locked to each other perfectly in time. You have probabilistic ins and outs. Reasons that those regions will feed a signal to A differs moment to moment as well. But you cannot drive A unless you have things feeding into A. So in that way, local dynamics might be part of something like functional integration, or there's a common drive that allows that region to express its dynamics. So in this first paper, we saw several striking things, and one of them is a very close coupling between voxel-wise variability, here now the square root of the power, which is mathematically equivalent to the standard deviation in the same band-limited signal. So you can think of it like a same measure we've used in the past. Now this is at rest in the Rockland data. So what we measure in terms of integration is PCA dimensionality. And if you have a space-time matrix and you compute how many dimensions it takes to capture a person's brain, so we do this for every person, the lower that number is, the more voxels are talking to each other. Okay? So when this x-axis goes to the left, they are lower dimensional, and y is the amount of power expressed in those voxels. So if you take the Shire mass, we had 14 networks we looked at, and the peak of all of this relationship was in the basal ganglia thalamic network that Shire has. Something special about this, that the coupling between dimensionality and local fluctuations was maximized, and this system overall was a clear marker of whole brain fluctuations in general at rest. So if this system provides a key local variability-based signature of network integration at rest, and these regions are connected themselves via the pallidum or directly from thalamus to striatum, can we move this to the task domain? So can we find change-change relations? This is not just a trait effect, but we can move it under cognitive load, linking local and local dynamics and integration. And now coming back to the DA story. and behavior as well. What does that have to say in this space? So we have a pretty unique study called COBRA that's been collected in Umeå, Lars Nyberg's group, of 162 older adults with fMRI and dopamine PET. The first wave is in, wave 2 is near complete with 3 waves planned. So we'll just talk about wave 1 results today, where we measure this SD-bold per load level within person and network dimensionality the way that I described it, the number of components required to capture 90% of your space-time matrix in a number of networks. Yeah? So we're quite interested in the change domain in the 2 to 3 back scenario, and that's mainly because that's where most of the individual differences lie on this task. If anyone's used NBAC, you'll notice that what you see is some people in green, for example, will continue to ramp here in variability. We've seen this in the past in 2 studies, but there's a proportion of people that will hit a cliff there at about 2 back, and they just taper their dynamics off. So this is really where people diverge the most within person. So we're going to focus on that here. And in change, what we see is what we hoped, and people that continue to integrate from 2 to 3 back, so they do not hit this cliff in terms of integration as well. They're pushing towards an even lower dimensional solution within the thalamic striatal system, are those that continue to ramp their variability from 2 to 3 back. Okay? If we expand that simple change model to include these other variables of interest, striatal binding potential, with raclopride, speed, accuracy, interactions, and so forth within this 2-3 shift scenario, what we notice is that people that continue to integrate, as we just saw, are those that rise in variability, but that population is also those with higher dopamine who are also faster. And in particular, a continuous variable interaction between the shift in integration from 2 to 3 back and speed is present. So the action is that on the left, these people that continue to integrate on the x-axis, only when they are also fast are they the ones that rise in variability from 2 to 3 back. If they are missing one of those components, so even in the fast people, if they don't continue to integrate, variability is falling off. So quite some difference in that regard. So we could say, okay, that's been a very focal look at the BG-thal network system, but what about these other networks that would be obvious to look at in terms of a working memory task? So when other key networks become the DV now, so we look at shifts in variability in the FPN or the DMN or the visual network or the DAN, by their own within network integration level, et cetera, just as we ran the BG-thal model. Dopamine and performance in all of those models is very weak. There's no PCA DIM or integration by performance interaction present. And most importantly, if you add controls to those models, that the BG-thal network integration score is allowed to be a predictor as well. What proportion of the delta SD bold stuff can be captured by this, what we think is a central network integrator, and it uniquely predicts fluctuations in all 4 of those other networks, but the reverse is not so. Yeah? So those networks integrating do not drive BG-thal, but BG-thal drives those. Yeah? So what we see is that so far, dopamine is a core aging and cognitively relevant neurotransmitter for understanding how local dynamics and network integration trade off. Within person under load now, so beyond rest, the first work that we did, when the BG-thal system integrates, the brain fluctuates. Not just the network itself, but the brain. It's a central driver. So this idea of low-dim manifolds, Max Schein has very nice work. Kai Huang has really worked on the thalamus a lot as well lately. And I think the importance of these considering low-dimensional solutions for a brain now that is under load is an important next step. So for me, at least, it's a promising underlying basis for understanding why we have the variability effects we have when we study aging and cognition. And of course, the meat is always at the longitudinal level, so we'll wait for COBRA Wave 2 to say more. So if you haven't had enough of this topic, there's 4 posters in the group, Julian Kosiesa, Niels Kusterman, and Alistair Perry, talking about different aspects and ways to both measure dynamics in EEG simulation, but in fMRI as well. Niels moving the work to the norepinephrine domain, considering the pupil a lot, and Alistair working on network distributed links in this domain as well. So thanks to everyone involved with this work and all the funding sources that we have. Thanks for you coming today as well.